Good afternoon, everyone. Hopefully you can see my slides okay there. Okay, so just to kick off, first of all, for those of you that don't know about Fidra, I'll just explain that Fidra is a small environmental charity based in North Berwick in Scotland, and we are scientific evidence driven, and we have a vision of achieving sustainable societies and healthy ecosystems. And ultimately, we seek to tackle chemical and plastic pollution. We have a range of projects. So, alongside this sewage project, we have a project that tackles chemical flame retardants. We also have a PFAS project that is focusing on pesticides and PFAS in pesticides at the moment. And we have our well-known NERDL project, which is tackling microplastics. So we aim to be pragmatic and solutions focused. We collaborate with a range of stakeholders, and we like to try and influence positive change. So let's start by thinking about the sources of chemical and plastic contaminants in wastewater and sewage sludge. So this slide just lists some of the sources, and I'm sure many of you are aware of these sources, such as the microplastic, synthetic fibers that come off when you're washing your clothes in the washing machine, microplastics from personal care products, cosmetics, and one of the big ones, tires, the microplastics that come off tires and get washed down into sewers from roads. And unfortunately, because of the persistent nature of plastics, around 90% are retained in the sewage sludge. The sewage sludge being that byproduct, the solid phase after sewage treatment. And chemical contaminants, major sources of chemical contaminants of interest to Fidra, these sources are also, these types of activities are also sources of microplastics, it should be noted. So diffuse pollution sources like the runoff from industry and agriculture, and then the more kind of point source release of domestic and industrial wastewater directly to treatment plants. So Fidra acknowledged that sewage sludge, treated sewage sludge or biosolids that get applied to agricultural soils and also get used for land reclamation, restoration activities, are rich in organic matter and essential crop nutrients. So it makes them a really valuable resource. Especially phosphorus and nitrogen. Phosphorus and nitrogen concentrations can be around 10 kilograms per ton in biosolids, and that's a lot higher than the concentrations of these nutrients found in other organic fertilizers like your farmyard manure, for example. So they're rich in these essential nutrients and soil health enhancing organic matter. But the unregulated persistent contaminants are a concern to Fidra. They represent a problematic barrier to a safe, clean, circular economy. And we also believe that they're a threat to soil health. And I'll be presenting the research by James Hutton Institute that seek to address that. So as Fidra is particularly interested in PFAS and microplastics, this slide is just a reminder of the challenges associated with these groups in particular. So there's a growing body of evidence that PFAS and microplastics are linked to issues with human and environmental health. PFAS is a very complicated space with respect to regulatory complexity at the moment, but it's a largely understudied chemical group that has been proven to have negative effects on both people and wildlife. And microplastics, due to their persistence, of course, are also frequently mentioned in media. They are also problematic, not just because of the persistence, like with PFAS, they can absorb other chemical contaminants. So chemical contaminants stick to the surfaces of plastics, and then those plastics act as a transport mechanism. So we see contaminants that enter the terrestrial environment are they reach the aquatic environment. And the question that Fidra wanted to get robust evidence to help try and answer was whether contaminated biosolids really do have negative impacts on soil health. And that's where we approached Rupert, because we were aware of the 2018 Scottish Government commissioned research that Rupert and others carried out. And we were keen to get an update of the risk analysis component of this research in light of new evidence that was coming out from published research. So we wanted that new data and evidence to be utilised to reassess sewage sludge risks to the environment. And we wanted to focus on the microplastics, organics, pharmaceuticals, personal care products, rather than the regulated metals and the pathogens that are controlled on a voluntary basis at the moment. And we do actually welcome improvements that we hope will come actually in regulating pathogens as well as metals in sewage sludge in the future. But we believe that we do need to keep a watch on these other categories of contaminants that get overlooked. So the approach that Rupert used to assess risks from contaminated biosolids to soils used these 4 steps essentially. First of all was the hazard identification. So identifying and prioritizing the hazardous agents. Of course, microplastics is more straightforward, but the organics and the pharmaceuticals, there's a whole range of different chemicals out there. So they had to be prioritized. The second step was the exposure assessment. So estimating the concentration of these hazardous agents in soils following the application of biosolids. And it should be noted that the concentrations within the soils themselves, so the receiving soils, were not taken into account. It was just the contaminants within the biosolids, using robust data in terms of typical biosolid concentrations that could be expected for the UK. So yeah, the exposure of the soil biota to these hazardous agents was investigated. And then something called fugacity modelling, which is a way of partitioning contaminants based on their physical chemical properties and predicting whether they'll be found in the soil, air, water, or sludge amended soil. Step 3 was the dose response assessment. So that's evaluating the toxicity of the hazards or the hazardous agents using predicted no effect concentrations and other metrics for soil biota. And that's where published toxicity data was used and assessment factors were applied to account for uncertainty. At this point, there was probably cases where some of the chemicals of interest did not have the ecotoxicity data available to do in assessments and so on. So that should be borne in mind, that there's certain substances that we don't know enough about. But for the risk characterization, the most important step, that was where the predicted exposure levels were compared to the toxicity thresholds. So you could think of it as taking the predicted effect concentrations and dividing them by the predicted no effect concentrations to get a ratio or a relative risk. And a relative risk value of greater than 1 indicates that there's potential for adverse effects. Okay, so moving on to the results, starting with the microplastics. So this figure here shows the relative risk or risk ratio for microplastics based on a single application of biosolids. But you'll see that there's a range of application rates that are considered. And there was no perceived risk after a single application. And I should also note that the green and blue lines represent the different methodologies that are used to determine microplastics in soils and biosolids. So microplastics can either be measured on a mass basis or based on counting the numbers of microplastics using microscopy. So we needed to account for the two contrasting methodologies there. But the important bit, when Rupert investigated repeated applications of biosolids and the risks to soil biota from microplastics, the research showed that repeated applications posed an impact or adverse effect on soil biota. And again, looking at the different ranges of application rates, you can see the changes in relative risk. And moving on to the organic contaminants, this figure here shows the partitioning of organic contaminants when after the biosolids is applied to the soil. It's like a measure of whether those contaminants will go into the soil poor waters or the soil poor air, or be retained in the solid phase, the soil and sewage sludge. And you can see a lot of blue and brown on that diagram. So it shows that these chemicals of interest that were prioritized for investigation showed a particular kind of desire to be found in water and the solid phase. And then if we look at the relative risks to soil biota from these organic chemicals, note that this diagram is using a logarithmic scale, because of course there's a whole range of chemicals and a whole range of values. So it was to simplify things, but a value of greater than 0 in this case meant that further investigation was required because there was a potential for risk to soil biota. And you'll see that there's 10 chemicals there highlighted red, and there's PCBs in there, and there's also the PFAS, the PFAS that is regulated, and there was plenty of data available for those PFAS, PFOS and PFOA. And it should be noted that a 2019 report that was commissioned by SEPA did actually find that PFOS didn't pose a risk to the environment, but since that report was published, the PNEC values for PFOS have since been revised to be lower. So it's likely that it would have been found to be a risk to the environment if that research was repeated again. So it's worth bearing that in mind. Also, there's a flame retardant there, TDCPP, that's just below that threshold of the risk ratio. So there's something to be said for continually us needing to review risks based on new evidence that comes to light. And moving to the pharmaceuticals and personal care products, again, looking at partitioning of these contaminants that were selected for investigation. Again, lots of blue and brown. So these contaminants were more likely to be found in the soil pore water and the solid soil and sewage sludge phases. And of course, these properties are used to, model the risks. And for these pharmaceuticals and personal care product contaminants, the relative risks, again, there were 10 chemicals that were identified as requiring further investigation. And interestingly, when we look back at the 2019 CEPA Commission's report on organic contaminants and pharmaceuticals, there was a pharmaceutical called galaxolide, hopefully I've got that right, which was identified as posing a risk to the environment, but that compound couldn't actually be assessed in this research because there wasn't enough data available to include it in the assessment. So that's another thing to bear in mind, that when looking at the specific risks to soil biota, we needed this specific toxicity data to do robust assessments. So just to summarize the results here, with microplastics, we know that they build up in our soils and they pose a risk to soil health. Just a few applications of biosolids could lead to, problematic microplastic pollution. With organic chemicals, it was found that several of the chemical contaminants found in biosolids can pose a threat to soil organisms, and that's after a single application. So of course, bear in mind that this research was just looking at a single application when it came to the organic chemicals and the pharmaceuticals. And PFAS showed significant potential to accumulate and threaten soil health in the long term. Pharmaceuticals and personal care products, almost half of those investigated posed a notable risk to soil health after a single application. So something else that really needs to be mentioned here is that this research couldn't take account of the poorly understood cocktail effects. So the research here considered each chemical in isolation of each other, but the application of biosolids to agricultural soils introduces these chemicals as a complex mixture or a complex cocktail. And it's believed that these mixtures can have a synergistic effect on soil biota. So more harm can be done to nature than the individual chemical when they're present as mixtures. So yeah, the combined impact of contaminants on soil ecosystems could be more significant than the additive effect of the individual compounds alone. And I guess in the future, to try and address this poorly understood cocktail effect, we need the mixture risk assessments, like improved modelling approaches. But just to summarise recommendations that were made by the James Hutton Institute based on this research. Firstly, adoption of the precautionary principle, improved wastewater treatment, increased research on emerging contaminants, improved monitoring, alternative treatment technologies, enhanced quality standards, and of course, improved agricultural use of sewage sludge policies. And to conclude, the reassessment of the environmental risks of sewage sludge use in agriculture highlights significant concerns regarding the accumulation of contaminants such as microplastics and PFAS, in particular, agricultural soils. So just to say on top of that, Fidra seeks to also see in the future improvements in UK and EU regulations, because we believe that at the moment biosolids are not managed in a completely safe and environmental beneficial way. So we welcome any improvements in the powers that regulators can be given when it comes to controlling contaminants in sewage sludge. And I'm sure water industry would also welcome the upstream wastewater solutions. So that would be commitments from across the biosolids supply chain stakeholders to prevent contaminants of concern going into the sewage sludge in the first place. And that would need, strong public commitment as well. And it's good to see that in the EU there is action under the Urban Wastewater Treatment Directive to apply EPR, extended producer responsibility schemes for pharmaceutical and cosmetic producers. So under the polluter pays principle, they will have to pay money towards the kind of cleanup or treatment of wastewater. Fidra also welcome technology developments that would provide downstream biosolid solutions to remove contaminants from the biosolids so that they're safe and clean to support a circular economy. But we acknowledge that those technologies aren't quite there yet. And we know that there's a lot of researchers and industry investing in technologies to be able to remove things like microplastics and PFAS. We also ask that harmful substances identified require testing and just ensure that the thresholds reflect best available science, so they get updated as appropriate. And we know that the EU sewage sludge directive is looking to review the level of contaminants in sewage sludge that are covered by the regulations. Welcome tightened soil protection, improved sampling and monitoring for the biosolids as well as the receiving soils. And we're aware that there are the chemical investigation programme, the SIP4 research trials that are underway, and we would be really interested to see the PFAS results from that research that's being done by water industry. And finally, we welcome any regulatory action that could be taken to support the use of the precautionary principle until the biosolids can be proven to be a clean, safe resource. So I'm now going to move on to our farmer survey findings. So we got 54 farmers to participate in a short survey that we designed to gauge general farmer opinion on the use of biosolids and other organic fertilizers or bioresources. So we found, well, there were 40 participants in England, so mostly it was based on English farmers, but 25 out of the 54 had previously used or currently use biosolids on their land. So that was useful to know. And in terms of factors influencing choice, things like organic matter provision, good use of nutrient-rich resources, as well as price and availability were factors that influenced farmers. And of course, over the years, we've seen big increases in the cost of synthetic or inorganic fertilizers. So it's understandable that farmers would look for other sources of nutrient provision. Farmers were aware of the potential presence of contaminants within organic fertilizers, bioresources, and they were also aware of the need to treat sewage sludge before application to land. So things like the biosolids assurance scheme, the safe sludge matrix, they were aware of that. And it was the metals that they were particularly aware of as well, metals and pathogens. So many farmers were aware of contaminants, perhaps not so much the persistent organic chemicals such as PFAS, but definitely the metals and also pathogens. And the farmer survey findings overall told us that 89% of participants were concerned about the potential risks from the application of contaminated biosolids, as well as other kind of more industrial organic fertilizers to land. And by industrial organic fertilizers, I mean things like the food waste composts. And 84% of participants would welcome tighter regulation and monitoring of biosolids and other organic fertilizers for contaminants. And I just want to point out that we are aware of the certification standards, the PASS certification schemes for digestates and composts, which we know help to maintain quality in terms of things like microplastics. And it would be great to see more in terms of improving the quality of biosolids. So finally, looking at some farmer case studies. So we've published a couple of case studies on our website from farmers based in Fife. So, for example, Doug, who promotes soil health through agroecological farming practices. He uses the advice of experts to avoid composts that are contaminated with microplastics. And he believes that the use of sewage sludge on farmland is a big issue for the environment, as the contaminants within the sludge can travel to the wider environment, including watercourses. And Philip, again, a regenerative farmer who seeks to enhance biology by eliminating chemical inputs. And he recommends the use of cleaner on-farm composts, and he has his own like on-farm compost systems that he's testing out. And he believes that some farmers continue to use sewage sludge due to a lack of knowledge of the long-term effects, but also possibly price incentives. So that confirms, the things that we drew out from our survey of farmers. And we're going to continue to work with other farmers that we're talking to, including farmers that use biosolids and see benefits from biosolids to their soil structure, because we do need to appreciate all farmers' needs and viewpoints on biosolids. So also want to point out that Fidra appreciate that contaminants found in treated sewage sludge is just one of many anthropogenic activities that can release contaminants into soils and the wider environment. We know that contaminants make their way from terrestrial to aquatic environments. And like I mentioned earlier, we have a project focusing on PFAS in pesticides. And we are keen to try and improve tackle the contaminants in agricultural inputs as a whole. And we're also aware that there's no river in England that has been found to have a good ecological status. So we really need some positive changes to try and restore biodiversity and the health of our ecosystems. So finally, next steps in terms of Fidra's work, we the kind of soil legislation to be prioritised so that we can protect soil health. And I know that the Scottish Government does have a kind of roadmap underway to try and achieve soil protection and to eliminate risks to soil health, based on growing scientific evidence around potential impacts that contaminated agricultural inputs can have. We need to take account of these, and I know that the Environmental Standards Scotland recently published a report on the risks to soils in Scotland. So it'd be great to see more being done in relation to soil health. And for biosolids and other bioresources, we're keen for them to be proven to be clean, safe resources to support the circular economy. And Fidra seek open dialogue with all stakeholders, even if it's trying to find out where there's gaps in evidence or where there's some promising technologies to try and solve these problems. We're really keen to talk to anyone that would like to work with us on our project. So I just want to say thank you very much for listening, and we'll take any questions that you might have.
Thanks so much, Jo. That was a great overview. I'd like to just check with Rupert first if you have any sort of additions you want to make or any comments you want to make before we move forward.
No, I think I'm just happy to hear what participants have to ask. That'll be fine.
Great. Okay, so if I can ask people if they've got questions to put those in the chat. I can't see any at the minute unless I'm not seeing a refreshed chat. Somebody let me know. So meanwhile, let me take the chair's prerogative and ask a couple of questions myself. I think one of the things you mentioned, Jo, was around chemical mixtures and cocktails, and perhaps both of you could talk about how we can improve our understanding of chemical mixtures, because to me that seems really important. If we are putting these mixtures, you know, they are present in the sludge, but we're assessing them individually, we're unable to assess them together, we're not getting a real world risk. You know, it's very hard to do that. So what are the options for us understanding that better?
Okay, I know that I kind of alluded to the fact that we need modelling that's kind of based on like the risk from mixtures, and Rupert would probably know more about the status of these kind of risk assessments being developed. But we know that when chemicals mix together in the environment, they can cause even more harm to nature, based on research articles that are out there. And there was a recent article on PFAS actually that indicated that the PFAS in biosolids was the impacts of PFAS in biosolids was exacerbated due to the mixture effect, and that was harm to human health. And It was a study by the State University of New York, I believe.
Great, thanks, Jo. Rupert, have you got any thoughts about the mixtures, the cocktail effect, and how we understand that risk better? It's kind of difficult thing to do.
It's a very difficult thing to do in the sense that whenever you do any of these types of assessments, your enemy is uncertainty. I mean, it's one reason why we set assumptions to be very precautionary when we're doing these assessments, is to try to override as much of the uncertainty in each steps as possible. As soon as you have a mixture that might interact in certain ways that you don't know about, there's vast amounts of uncertainty to account for. I think probably the most pragmatic way to deal with that is to actually undertake more ecotoxicological studies on products as opposed to on specific chemicals. So I mean, it's one thing you don't really find so much of in the literature is like we took these different types of compost, for example, and applied them to this and these soil environments, and these were our results, sort of thing. It's always measuring specific chemicals, which it makes it very hard to look at anything other than individual chemicals.
Yeah, brilliant. Thank you so much. There is a comment and a question in the chat, so I'll just read those out. So there's an offer from David Ray. He is working, he's from an activated carbon company, and they're working with other companies regarding the abatement and destruction of PFAS and POPs, and he said he's happy to share current best practice and latest developments. So thanks for that, David. I'm sure we'll be in touch later. And it was really interesting to see from the water companies just last week, Water UK, which is a membership organisation across the UK, Scottish Water and all the England and Welsh water companies, sent a letter demanding better source control of PFAS. So it was interesting to see water companies in the UK coming out on that. There is a question from Julia Sussons from DEFRA. Could you explain a little more about the data and methods used for the exposure assessment and risk characterization? For example, the variety of types of polymers, size and shapes that make up microplastic emissions to the environment present a variety of ecotox risks. How did you tackle that? So that's quite a technical question. Rupert, do you want to give that one a go?
It's quite a technical question. I mean, you see very useful studies that combine different ecotox estimates, which is very useful for me anyway. So, for example, you might have a study looking at fibers and their impact on, let's say, worms, for example. And you might have another study that has, particular sort of granular sizes of, microplastics and their impact on a different organism. What happens in I suppose simple terms is there's studies which look at all the different studies published, and then they combine those statistically in order to get an overall estimate of if a variety of microplastics are put into this environment, what would be the impact on biota. And there's some of the studies that we certainly relied heavily upon. So they basically run all the different PNEC values together, the no effect concentration values together. They combine them statistically and come out with a this is a typical value. This is a sort of a low value that when we adopt the lower values because it's more precautionary. You see what I mean? So they would say they might take a 5th percentile value or something like that. But yeah, there's ways where these different studies are combined, but certainly within the data that we used, we had data on fibers, data on granules of different sizes, from larger ones down to very, very small, sometimes called nanoplastics, that are thought to get inside different structures within organisms. So The whole range was sort of incorporated into the data, but ultimately these studies will combine in order to get a single value, which, there's pros and cons with doing that. And again, by keeping it precautionary, we try to override all that uncertainty that's in it. So I don't know if that's an adequate answer. I'm not sure.
That's great. Thanks, Rupert. And I think, if you've got any further questions, Julia, do just email us and we'll see if we can get back to you directly on that in a written answer if you've still got some queries. I can see that we have a couple of hands up as well. I'll just take one more question from the chat and then we'll go to a couple of hands up. So Rachel Salvage is asking around flame retardants, and are all of those that were found deemed to be of low risk?
We didn't have any flame retardants that presented an appreciable risk with this assessment, no. But there were some flame retardants included. Yeah.
Great, thank you.
Just to point out, we did make an effort we asked Rupert to look at specific ones that we were keen to know more about.
Yeah, I think one of those was sort of just below the threshold. Is that right?
Yeah.
Yeah. So if somebody could unmute John so that he can ask his question, John Redshaw.
Yeah, can you hear me all right?
Yes, we can, John. Nice to see you. Hi.
Great. Thank you very much. Thank you for the invite and thank you for an excellent talk and research study, Rupert and Joe. A couple of thoughts are coming to my mind as we were going through the talk. I think we all probably recognise this is clearly part of a much bigger jigsaw, isn't it? That this issue is a One Health issue that recognises the links and interdependencies between humans, animals, plants, food and the environment. So as we think about these chemicals in soils, we inevitably think about potential for uptake into the food chain, into humans, etc. Just a few other comments before a specific question. We also recognise that, and I appreciate you said you weren't thinking about pathogens, not at the moment, but as part of a wider agenda, but that microplastics will act as a substrate for the development of biofilms, and through that for the exacerbation of antimicrobial resistance. We also recognise that many chemicals exacerbate AMR in the environment through co-selection, and that people are starting to derive PNEX for AMR, just as we've got for ecotox, and that would help to, if you like, develop the risk assessment approach. But as we think about mixtures as well, this is something else that was going through my mind. And I think perhaps one question, firstly, which were the most sensitive species did you find when you looked at the species sensitivity distributions for these particular contaminants? Did you find particular species being more impacted than others? And if so, does that lend itself to the development of a more direct biological effects-based tool for future assessment and management that takes better account of the chemical and ecotoxicological interactions? So I don't know if there were particular organisms that were more sensitive, and whether in these days of genomics and molecular biology, you feel there's scope for having a more direct biological effects-based tool to aid in these assessments.
Yeah, I think the scope, John, definitely. It is possible, and certainly the studies that I mentioned earlier where they combine lots of different PNEX, they will rank the different organisms in them in terms of their sensitivity. What I wouldn't say is that from what I was looking at, there was any particular organism that I felt in different studies was always standing out as the most sensitive. I wasn't specifically looking for which is the most sensitive organism when I was doing the work, so that would be one I'd go back on and have a look at. To be honest, in terms of that, because that wasn't my aim when I did the work. But yes, certainly there are studies out there that have a genomic basis that are ranking the organisms on the basis of outcome. Yeah, that would be an interesting point, I think, to maybe refine how you might manage some of these things.
I think that's very helpful. I'm certainly aware of work under the NERC Freshwater Quality Programme, for example, the project, the Pacific project, that is looking at microbial molecular approach and developing, if you like, signatures for different chemical cocktails. So those sorts of things are quite attractive. There's also the Environment Agency is putting a lot of effort into looking at biofilms on stones in rivers and the microbial communities with them, again, with a view to providing, if you like, a more direct biological effects based approach for the assessment of complex mixtures of chemicals and their interaction with environmental stresses, because this is one of these old chestnuts that's been with us for decades.
Yeah,
is how do we best deal with this? And I think the obvious thing in my mind is to have that part of the triad completed. So we're not just looking at chemistry and ecology, but we're starting to link the two together through biomarkers and bioassays, etc., that provide a more direct expression of exposure and impact.
And I think that's
thank you so much, John. Rupert.
No, I was just gonna say yes. I agree. That would be a really good area of development. Certainly supportive of that.
Yeah, great. I'm just going to go to a couple of comments in the chat, and then I'll go to another question online. So Sharon has kindly shared some research on mixtures and their impact on fresh water. So there is a link in there, so we can have a look at that. Alan Cundhill has asked, what alternatives do project authors feel could be deployed for the management of sewage sludge if use on land needs to be reduced or stopped? Or is it a case of improving treatment of sewage sludge and reducing contaminant inputs to the wastewater system to improve safety on sludge use on land? Jo, do you want to take that one?
Yeah, so I guess my slide did address that with the upstream and the downstream solutions that we'd welcome. But if we were to reduce the amount of sewage sludge being applied to land, the alternatives would be the extraction of nutrients and the use of pyrolysis. Unfortunately, I know it's a high energy like destruction of the sewage sludge, but pyrolysis can be used to remove contaminants like PFAS. Not all PFAS potentially, but I believe there are some technologies out there that claim to be able to remove all PFAS from biosolids through pyrolysis. But then the product of that is biochar. And I wouldn't want to advocate the application of biochar to agricultural land. I think it would be far better to use those kind of wastes of minimal kind of value, use them in making bricks or building materials and so on. But unfortunately, incineration is a route that countries like Germany are using. So I think we can't lock ourselves into one route, but ultimately we need to try and address the contaminants in sewage sludge and stop causing harm to the environment from contaminated materials that go.
Thanks, Jo. How does the UK compare to other countries? Just out of interest.
Yeah. Oh, I do actually have a slide. I would need to share it. We have a slide, but I can click through to quickly. So for example, this shows how the UK is, you know, quite a high user of sewage sludge in terms of applying to agricultural land. And then you can see other countries that apply to land, and then those that avoid it or don't apply as much.
That's interesting. Thanks so much, Jo. And I hope that's answered your question, Alan. So I'd just like to move to a question on the chat, so we can allow Jamie Page to ask his question.
Yes.
Hi, Jamie, please go ahead.
Yes, hello. Yeah, I had to unmute myself too. Yes, well, thank you very much for this very interesting presentation. I'm from a health charity, the Cancer Prevention Society, but I am equally concerned about what ends up in the wider environment because everything comes back in the end. Definitely in favor of upstream solutions. If you don't put the toxics into the system in the first place, you don't have to deal with them later on. And I think we need to have much more focus on, for example, the halogens, which tend to give the general problems of persistence. There's a focus on fluorinated compounds, but of course chlorine and brominated compounds. We have a particular focus on flame retardants, our organization. So if we avoid certain chemistries, particularly the group 7 elements, then a lot of these problems, I think, would be reduced. So we need much more holistic approaches to what we allow in. I wanted to make a comment about PNEX. One of my concerns about PNEX is many chemicals have quite high PNEX, but if you sum total, for example, all the organophosphate flame retardants that are going into the environment, you might be pushing over the PNEX for individual ones. That's one comment about PNEX. The second comment is the PNEX are often quite high, whereas I see in the scientific literature, I monitor on a weekly basis the scientific literature, and you see a lot of chemicals exerting effects at concentrations lower than the PNEX, often an order of magnitude lower. And so that then begs the question, does that mean we're going to allow river concentrations of these chemicals to bump up until they hit the PNEX before we do anything? When we know from lab studies, say of zebrafish, that they already have deleterious effects at concentrations order of magnitude lower. So we need to be careful of these PNEX, and we need to be, there are good databases like the Norman database, but we also need to be mindful of what the scientific literature is reporting. So that's my comment about PNEX. Thank you very much.
Thanks, Jamie. That's a really helpful comment. I don't know if any of the panel want to come back on that, or just happy to listen to that very valuable comment.
I can give opinions on PNEX. That'd be useful. I mean, like all these measures, there's they come with uncertainty and variability, and you will find studies which will be higher, studies which will be lower. So the main approach we took with the PNEX was to adopt the ones that had come from significant review and been, I suppose, adopted by agencies such as the European Food Standards Agency. So these groups, they'll have reviewed lots and lots of different PNEC estimates for a particular chemical, and they will have come to a conclusion over what an adopted PNEC should be. And obviously that's going to be a mixture. It is going to be a mixture of scientific evidence and also political and social aspects as well. But that was our approach, to use where as much as possible ones which had been agreed and adopted by national or international agencies. So yeah, it's a source of uncertainty in any analysis, that's for definite.
Yeah. Okay, great. Thank you so much. We're running out of time to answer all of these questions. I will try and squeeze another couple in because we do have so many. I do apologise to those we can't get to, and we will try and provide some written responses to those questions in the chat we don't get to. I think there's a quick question here about, do we have any information relating to the time taken for chemical contaminants to break down? So that's looking at the persistence. Do we know how long these chemicals take to break down?
We know from PFAS. Oh, I'll let Rupert answer.
Okay, I was going to say short answer, yes. Some of them break down much quicker. Some of them break down into different products that they themselves have a particular chemistry that you may or may not be worried about. Some of these chemicals, like the PFAS, they get labeled forever chemicals because although they do break down, you're talking lifetimes to actually fully break down out of the soil. So the different rates of decay were taken into account in the analysis for certain. But, you know, when we're assessing things like PFAS, we just don't assume any breakdown because there's no reasonable timescale there.
And we should maybe mention, we know that with PCBs, we've seen, due to the legislation change, PCB concentrations have kind of improved over the years. But of course, we still find them because they're persistent, and the PFAS problem will be with us for years to come if they keep accumulating. And we know that PFAS can break down into more harmful PFAS as well. So,
yeah. Okay, great. I'm going to let Tim just ask his question so that we do get to hear it. And then I think this will have to be the last question because we've reached the end of our hour. So Tim, if you could keep it short, that would be great.
Yeah, will do. Thank you very much for the presentation. Really enjoyed it. I just wanted to ask a question about the methodology of the experiment and whether you used control sites and whether you could be confident that the substances you're seeing within the soil were from the biosolids, or whether there could be other sources like atmospheric deposition or other materials spread on land. So yeah, that's my question. And I just lastly wanted to ask whether you also considered human health risks, you know, those onward risks in the food chain from crops grown on land that's been spread with biosolids. That's it from me. Thank you very much.
Thanks, Tim.
So that's a good question. I mean, this wasn't an experiment, just to make that clear. So this is a theoretical exercise of, if you put sewage sludge onto soil, what might be the impacts? So we were only interested in what has been measured in sewage sludge. So we searched the records for measurements of chemicals in that sludge, and then we theoretically say it's applied to soil, and these might be the impacts to the soil biota. So in that regard, we aren't thinking of any of the other inputs, and there could be other inputs. You know, depending on what location, you know, if you're actually talking about a real location, there could be all sorts of things going on. The 2018 report looked at the human health impacts from sewage sludge to land, and this most recent report just looks at the impacts to soil biota. So it's two different endpoints. I mean, what I would say is I think both need updating, and I'd also would support in what Joe said about creating a quality standard. I think that's when we did very similar exercises for compost and digestate, which led to the development of the PAS 100 and PAS 110, and I think something similar for sewage sludge to enable it to be used in a consistent manner, I think would be a very sensible route. But yeah. But I'm a scientist, I shouldn't give such opinions.
Thanks, Rupert. Joe, do you want to come in?
Yeah, thanks, Rupert, and just to confirm that actually I think we need to keep a watch on contaminants and consider revisiting the human health effects, and that would help with John Redshaw's AMR concerns and the biological cocktail effects as well, potentially, as just as important as the chemical cocktail effects.
Thank you.
That's great. Thanks so much. So like I said, there are a few questions that we didn't manage to get to in the chat. I do apologise to those who put them in there. We will, I promise you, get back to you individually on those questions. And to remind you that we will be able to send a link of this recording around to you after the webinar. I wanted to give Rupert and Joe just a sentence or two each if they want to sum up. And I wanted to thank the speakers, attendees very much for today's useful presentation and discussion. So Rupert, did you want to have a last word or thought or reflections?
I just want to say thank you. It's nice to be able to actually have a discussion about something like a report like this with obviously interested parties. It's been helpful and made me think about a few additional things as well. So that's good. Thanks,
Rupert. Joe?
Yeah, just say thank you as well, and also to say we're looking forward to seeing changes in legislation and regulators being having more regulatory powers to enforce, improved practices, let's say, with sewage sludge use. Thanks.
Great, thank you so much. Thanks everybody. Take care. Bye bye.
Thank you.